Welcome to the second episode of Innovation Matters, a podcast organized by the Netherlands Innovation Network. This episode will continue to talk about antimicrobial resistance and is broadcasted from Shanghai, China. In our last episode, we spoke extensively about the problem of antimicrobial resistance, also called AMR. Antimicrobial resistance happens when microorganisms, such as bacteria, change when they are exposed to antimicrobial drugs, such as antibiotics. Together with Dr. Maarten van Dongen of the global information platform AMR Insights, we spoke about the different sectors involved in the problem and about the 5 strategies on how to curb AMR. We spoke with Dr. Yonghong Xiao, co-writer of the Chinese AMR Action Plan, about China's goal of developing new innovative drugs, like new antibiotics or alternative treatments like phage therapy or protein drugs, by the end of this year. This episode we will go more in depth and talk with 3 different researchers who all tackle the AMR problem from a different perspective. We will talk with Nathaniel Martin of Leiden University. He's a professor in biological chemistry, and with his research group he tries to understand bacteria at a molecular level. This should help in designing new antibiotics and in developing new molecules that will block resistant pathways bacteria have developed. We will also talk with Professor Arjan de Visser, evolutionary biologist at Wageningen University, who is working on models to predict evolutionary pathways. He tries to understand the determinants of evolution and by doing this get insights in why bacteria evolve resistance. But we will start with our first guest, Sam Linsen. We will talk about antibiotic pollution in our environment, in rivers, our drinking water and milk. Surprisingly, this is not yet measured adequately, while mapping the issue is a key part of finding a solution. Sam Linsen is a Dutch researcher and started his business Squared End in China, which focuses on antibiotic residue testing and data gathering in order to map the extent of the problem worldwide. By gathering enough data, his company should be able to track sources of antibiotic pollution in our environment. Welcome, Sam Linsen. Could you please briefly introduce yourself?
So my name is Sam Linsen, and I'm the founder of Squared End. It's a Chinese company that measures the antibiotics in food, water, and the environment. And in fact, we are a technology company that's developing a platform to test for antibiotic residue in food and in water, in order to inform the public about antibiotics and antibacterial activity in their environment.
So you're constructing a platform that should give an overview of the antibiotic residues in the products we consume on a day-to-day basis, but also the residues in our environment.
Yeah, so in fact, it's mostly about antibacterials, antimicrobials that are, for instance, used in food production. And then we specifically think about animal protein production, which could be meat, which could be shrimp, which could be fish, which could be milk, which could be eggs, because the animals that produce these products, they often receive antibiotics during their life in order to treat, of course, infectious disease, but also to prevent infectious disease.
Is the use of antibiotics in livestock the main cause of the pollution of our environment?
There are many sources of antibiotics in the environment, and animal food production is one of them, but definitely not the only one. Antibiotics actually come from microbes, because in the end, microbes produce antibiotics. They produce antimicrobials. They have been doing that for billions of years.
Okay, hang on. Maybe we should go back to basics here first. So antibiotics are actually a natural compound.
Yeah, I think that's a little bit of a misunderstanding, and I think it's also not completely solved. What actually is meant with what definition. But I think in general, you could say that there's a family of antimicrobials, and antimicrobials would target microbes, right? So microbes is actually also not a very well defined group of organisms, but in general, we think that viruses and bacteria and algae could be, in a microbe family. Well, if those microbes, they produce the antimicrobials themselves, then we talk about antibiotics. So antibiotics are just natural products that inhibit the growth of microbes or kill the microbes.
So antibiotics are naturally present in nature, but the pollution is a result of human misuse. You mentioned livestock as an important source. What other sources are also contaminating the environment?
Yeah, so these antibiotics, they in the bigger countries that produce antibiotics, like the active pharmaceutical ingredients, it is likely that some of those traces would come from those factories. Those producers are then mostly located in India and in China, and these producers, they run factories, and up to 50% of the ingredients they produce end up in the river because of the waste water production that they have and the fact that they do not filter very well. So that is then, of course, looking at the human pipeline, that is the first point where antibiotics end up in the environment. But then, of course, we also have the food production, but also don't forget the medical sector. Where a lot of antibiotics are used in many countries. We also look at pet animals. They use antibiotics. Yeah, so everywhere where there is use or production, there will be pollution.
Do we already have an idea of the extent of the pollution?
Yeah, well, I mean, if we have a decent idea on the scale of the problem, I would say yes and no. Because let's look at drainage systems, right? If you look at lakes, rivers, and coastal areas, you can actually get a good idea on how pollutants in general spread. You can see some of the dynamics. For instance, if they are available to other systems. So for instance, if you have pollutants in one area, which is the source, of course, at that source. It may have a certain effect, but if the same pollutant then ends up in coastal areas or wetlands, it may have a different effect. So that's why it's not only of importance to track, only measure at the source, for instance, because many antibiotics may actually just bind themselves to clay or to stones and just remain there for a long time. But like a certain part of those waste products will also find their way down the rivers, the lakes, and the coastal water. So if you look at those drainage systems, then you actually see that basically in Asia and in Europe, in United States, a couple of examples in South America. I mean, basically there's not a single river, like a main water body, that does not have antibiotic residue in it. Of course, in those huge water bodies, the concentrations are very, very low. We always try to measure in nanograms per milliliter, or maybe even picograms per milliliter, which are extremely low concentrations, like 1 in a billion or less. But if you would actually calculate that into like an item that you would recognize, which is a pill, like a pill of ampicillin that you would take of 500 milligram, then for many of those main water bodies, like big rivers, it wouldn't be strange to find about 1 to 10 pills a second floating by. So this is just free floating antibiotics.
That is very impressive if you put it like that. So this is a measure of the antibiotic levels in rivers, free floating in our rivers. Can we also measure how much our, let's say, unintended antibiotic intake is that we consume through our food and our water? Can we say, for instance, on a yearly basis, we consume X amount of pills of antibiotics?
Yeah, that's a very interesting question, and I think that also comes closer to actually what I'm doing. Even though there's, of course, also no simple answer to that. Like the overarching answer would be, of course, to measure. And I think especially these days, every sane person on the planet would actually appreciate that measuring is a start, like of making good decisions. But then, of course, it's the question is how to measure, right? Because we don't want to be like this measurement to be intrusive. But having said that, there have been some groups, including one here close to where I live, in Shanghai, that have been following, for instance, school children and measured how much antibiotics they had in their urine. And they found that actually the exposure of antibiotics was relatively common for these school kids. So I think about 74% of these kids had antibiotics in their urine, but definitely not 74% of those kids actually was on an antibiotic regimen during the measurement. So where then the antibiotics come from, that's question number 2. It could be water that they drink or the meat that they eat, but this anyway could be a way to measure the exposure. So other ways, of course, are more you could try to build indices on antibiotics in the environment around you, where you try to generate an overview of course, the sources of pollution, the trace amounts you find in the water, and also for instance, the water that the animals drink, and maybe the urine that animals produce, that in the end will reach the consumer market. And by such an overview, I think it would also be possible to fine tune some of the risks, and also inform the public more to push them into a direction that they actually start to be more demanding on antibiotic use in the products that they use and eat in their daily lives.
And you are working towards that last strategy you mentioned. So next to that platform that you're building, you're also working on developing a test, a so-called chip, to measure the levels of antibiotics in fluids. And how does that test compare to current tests that are already available?
So what is my solution then? What I built is a platform to measure antibiotic residue, primarily in water. We also work on milk, and later we will also go into other fluids. And if we are possible to also build a chip, we also bring a solution that actually consumers can use themselves to test for antibiotics in whatever they buy. So this is something that can be done in many different ways, and has been done by different companies all over the world. Successfully to, for instance, help the industry to remove or to measure antibiotics in the dairy industry before milk goes into all kinds of production cycles for which you need bacteria to grow healthily. Yeah, so I'm also building such a tool, and the question for me was, with what is there on the market, can we actually maybe make an improvement step to add some value to the data itself that is produced by such a tool? Because what we see now is that you would get, for instance, you would get a tube from, for instance, the Delta test. You would add some milk, you incubate it at a certain temperature for a couple of hours, and then you read the color, and then you really get a yes or no answer. So yes, there are antibiotics, or no, there are no antibiotics. But it won't tell you what the antibiotic is, and it may also not tell you where it comes from. Because for every antibiotic for which the system that you test for is sensitive, you will just get one color. So I think, anyway, for antibiotic residue testing, there's a lot of room for different types of testing. Because we look at antibiotics, and antibiotics are defined based on their function, not so much based on their structure. And that means that to measure the activity, actually, you should measure for function. But if you're interested in just the presence or absence of a certain molecule, then you can actually test for structure, right? So that means that analytical techniques to test for them separate themselves into two. One is target one specific molecule or a family of molecules and say it is there, yes or no, and maybe also quantitate. And the other one is you just let biological systems like cells that you have in the freezer tell you if there is an antibiotic, and then by telling that if there is antibiotic activity in your sample, but you actually don't know what the antibiotic is. You cannot identify them. So I thought there is one thing missing here, where actually you can actually go into a sample where you know nothing about, and you do not make any assumptions. You want to test for antibacterial activity, but you also want to be able to say something about the specifics of that antibacterial activity. So ideally you want to also be able to identify those. But what if you cannot identify those? You can at least make the outcome of your assays unique in a way, or so diverse that different mixes of antibiotics will generate, different outcomes. So maybe then that is something that I developed then. We cannot necessarily identify each specific antibiotic, but we can measure antibacterial activity, and we can make the response unique to a certain combination of antibacterials. So we expanded, and actually we built a panel of all kind of like different type of sensors, and by that generate patterns. And patterns can, of course, be used for clustering. They can be used for source tracking, and by that, and of course, they can also be standardized. So we can actually compare, samples that are taken on different places or different times in the same or in different areas. Basically, I would say our platform would definitely be a data tool for which we built our own data collector. We can start to map whole areas. The delta of the Long River, or lake districts, or maybe areas around farms, together with people who are interested in keeping the antibiotic footprint of their activities low, or people who are interested to actually control the antibiotic input in, for instance, the ducks they breed or the cows they produce. And the same would be true for other food producers. All of this data then in the end is meant to inform the public about where can you expect to get safe and healthy produce, so how and you can actually base your purchasing decisions on that.
So the chip test that you are developing is more extensive in the sense it could test a wide variety of antibiotics instead of just giving you a simple yes or no answer to the question whether or not the fluid you're testing is polluted. And also the test data is used more effectively through data gathering, which in turn can be used to start tracing actual sources of pollution. I actually thought such platforms already existed.
So platforms to test for antibiotics do exist, yes. So they can be based on microbial inhibition. They can be based on immunoassays and ELISA, and they can be based on mass spectrometry. But there's no platform that integrates all these different types of data. And there's actually also no platform that allows the consumer to say, hey, let's compare my own footprint or fingerprint, so to say, with all the other fingerprints that are produced by other people. All the other techniques are much more vague and also won't steer you in the direction of where the residue that you find actually comes from.
So all current tools out there actually simplify the problem.
I know people just think very pragmatically, and I actually also talked to a milk farmer at some point, just saying if the level of antibiotics in my milk is too high, I just mix it with milk with less antibiotics. That is then a practical solution for a working class solution for a middle class problem. But of course, for the consumers, this won't. I mean, the consumer won't know. And okay, if the milk that they drink, which is not processed, apart from being pasteurized, does contain some antibiotics, they I think are much more concerned because they cannot mix it with something that doesn't contain antibiotics.
If I get this straight, we as consumers should be more aware of the consequences of pollution and urge our governments to take action. And then governments will be seeking tools that provide help, and Squared End, your company, could be one of them. Is that right?
Yes. Yeah, absolutely. I could provide like a standardized platform to sample like everywhere, basically, as a starting point for source tracking and defining like high risk areas.
What do you have to say to the people that are listening right now and want to take responsibility too? I mean, I would also like to demand more transparency for this. Is there somewhere they can go to or sign up? What if they would like to conduct the tests at home? What should they do?
Well, for me, I'm at the moment in the R&D phase, and it will still take some time before the products will become available for all the consumers. But at the same time, antibiotic residue pollution it's not something that will go away in the coming years. It's something that is here to stay. And therefore, I would like to invite you, the listener, to come to my website, squaredn.com, register yourself. So you will receive updates and insights on what we're doing at the moment. And whenever we're ready to start sampling in your area or do measurements, we'll get in touch, and then we will ask you to contribute with just sending some of the water in your environment to us, which then, of course, will be a huge impact for us, for our development, and also probably, hopefully, for the situation around where you live.
Thank you, Sam Linsen, for taking the time to talk about the development of your test and data company. So if you would like to follow Sam and support his journey towards more transparency, then please go to his website, squaredn.com, and subscribe. Like Sam said, the problem of antibiotic residue pollution will not go away, and mapping the issue is a start of the solution. However, as we already live in a post-antibiotic era, we do need other solutions as well. I'm going to talk about it with my next guest. My next guest is Professor Nathaniel Martin from the Leiden University, and he is running a research group which, on a very fundamental level, focuses on the resistance mechanisms that bacteria develop. Welcome, Professor Martin. Could you please introduce yourself?
Sure. My name is Nathaniel Martin. I'm a professor of biological chemistry at Leiden University in the Netherlands. And my background is actually as an organic chemist. I did a PhD in organic chemistry at the University of Alberta in Canada. And from there, decided to branch out beyond pure chemistry and look at the ways to apply it to infectious disease. And so that led me into the field where I now work, and that's trying to develop new antibiotics. And my background as a chemist is very influential there because, of course, as an organic chemist, one can understand things at the molecular level and try to develop solutions at the molecular level. So a big part of the research in my group in Leiden is designing new antibiotics to overcome antibiotic-resistant bacteria. We also try to develop new molecules that will block resistance pathways in drug-resistant bacteria. And if that's successful, then you can, in principle, resensitize drug-resistant bacteria to conventional antibiotics. So this is basically the two strategies we take in the lab, trying to develop new antibiotics or trying to disarm resistance in bacteria to overcome antibiotic resistance.
Let's assume not everyone who's listening has a background in biology. Could you please tell me more about what kind of mechanisms can bacteria develop to become resistant?
Sure. Yeah. So you have to envision bacteria as these small single-celled organisms that multiply very quickly, and they're actually all around us, and they're part of normal, healthy human life too. In fact, one of the startling statistics is that we have more bacterial cells in our body than we actually do have human cells. So we're actually more bacterial than human. And that speaks to how important bacteria are in our body, specifically in our digestive system. And this is the whole area of the microbiome you may have heard of. But what can happen is if you have a disbalance in the bacterial population in your body, or if you have an infection by a pathogenic bacteria that maybe gets in your bloodstream, then you have a problem. And these bacteria will circulate and of course, multiply and will make you sick, causing infection. And resistance to antibiotics takes many forms. I mean, antibiotics, when they were first brought to the clinic in the mid-1940s, really revolutionized the way that we could treat infections, because it was for the first time really recognized that one could take a drug that would specifically kill bacterial cells and not harm our own human cells. So it's the so-called magic bullet. And in the 20 or 30 years that followed the discovery of penicillin, or let's say the clinical implementation of penicillin, many different antibiotics were discovered, and many of them kill and target bacteria via different mechanisms. And in the last 50, 60 years, bacteria have begun to adapt and in doing so become resistant to these antibiotics. And you asked what type of resistance mechanisms are at play. Well, in some cases, bacteria can actively degrade or destroy antibiotics. They've developed ways of doing that. In other cases, they also have ways of spitting antibiotics out. So the antibiotic may get into the bacterial cell, but before it can harm the bacteria is able to spit it back out. And in other cases, bacteria are also able just to modify the target that antibiotics would normally hit, so that the antibiotics no longer are able to bind to the bacterial cell. So those are three main types of resistance, I would say. The destruction of the antibiotic, the spitting out of the antibiotic or the bacteria actually altering their own structures so that the antibiotics are no longer able to bind them.
And which resistance mechanism do you primarily focus on?
We focus primarily on two of those three. One major area is trying to develop what we call inhibitors. These are inhibitor molecules that can block the mechanisms that some bacteria use to destroy antibiotics. So if you have an inhibitor that blocks such a resistance mechanism, it prevents bacteria from destroying conventional antibiotics, and those conventional antibiotics in turn can then be useful again at treating these types of bacterial infections. So that's one line of work that we're actively engaged in. And then another one is the developing of new antibiotics, or let's say improved antibiotics that function again against bacteria that have altered their targets. And this is where a background as a chemist is very insightful, because if you understand at the molecular level how bacteria have structurally altered their targets, you can then in principle also look at the antibiotic, and in the lab we can alter the structure of the antibiotic to try and help it then overcome the modification that the bacteria has introduced in its target. So it's a bit of an arms race. The bacteria does one thing, then we do another to keep pace, and back and forth it goes.
Is it like changing the structure of antibiotics so the bacteria will not recognize it as its enemy and therefore will not attack it?
Exactly. Yes. This is a big part of it. So bacteria over years or decades of exposure to an antibiotic will develop ways, as you say, of recognizing an antibiotic as being something that's toxic to them, and they can then destroy it or block its action. So if we modify the structure of an antibiotic, as you say, we can also then, in some sense, trick the bacteria, or the bacteria doesn't sense the modified antibiotic the same way it does the original antibiotic.
If we are able to reverse the resistance mechanism of bacteria, does that mean that all bacteria will be susceptible again to all the antibiotics that became obsolete over time?
I wouldn't say that they would become sensitive to all antibiotics, but generally speaking, you talk about specific resistances to specific classes of antibiotics. And the other thing to remember again is, of course, bacteria adapt to pressures that we put on them, and antibiotics are a pressure. Now, if you have an antibiotic that's become resistant, or if you have a bacteria that's become resistant to an antibiotic, and we then can develop a new inhibitor that blocks that resistance mechanism and then resensitizes that bacteria to that antibiotic, there's nothing saying that the bacteria in response won't also evolve to become resistant to the inhibitor that we've developed to the first resistance mechanism. So you really do have to see it as a ever progressing or ever sort of changing almost arms race between these. But in principle, your assessment is correct. That is, if we develop inhibitors against known resistance mechanisms, we will be able to then once again use antibiotics that maybe haven't been very effective for the past few years. And that's also an important strategy and a viable strategy. There are, in some cases, clinically used combinations of what we would call older antibiotics plus a newer, more recently invented resistance blocking drug molecule. So these are established methodologies for certain types of resistance.
So even though your research is quite impressive, this is definitely not a silver bullet.
Yeah, there's no such thing as a silver bullet in the sense that it'll be forever a silver bullet. The best we can hope for is to have a bag of silver bullets, or to have an ever adapting and growing line of silver bullets, so that when one stops being effective, we've got a new one that can take its place. It's really the only way that you can ever really maintain a proper arsenal of antibiotic drugs, is to recognize that the minute we use an antibiotic is also the minute we start providing bacteria the challenge to evolve towards it or against it. And this is a completely normal process. I mean, Alexander Fleming, in his Nobel Prize speech, acknowledged that it's fairly easy to make bacteria resistant to penicillin. We can do this in the lab. We do this, in fact, when we work on new antibiotics. One of the standard tests we do is how easy or difficult is it for a bacterium to become resistant to this new antibiotic. And you do this simply by exposing bacterial cells to low levels of the antibiotic, so less than would normally kill the bacteria. If you do this over a period of days and weeks, you will often see that bacteria on day 30 now require much more antibiotic to be killed than they required on day 1. So this is something that is widely known in the field. It's, as I say, a natural process. So this is why also people need to recognize that even if we're very responsible with our use of antibiotics and limit how widely we prescribe antibiotics, it's still more or less inevitable that resistance will be developed. The best you can do is maybe hope to delay it as long as possible by prudent use of antibiotics. But ultimately, if we want to maintain our standard of medicine in the world, we also need to acknowledge that antibiotics and research in developing new antibiotics is quite foundational to being able to maintain our healthcare systems.
When can we expect your findings to reach patients?
Well, the standard number that you always hear in the drug development field is 10 years from the moment a drug is first made to the first moments that it will be used in a human patient. And that's, of course, because there's such a long and expensive trajectory when you talk about the sort of preclinical validation, demonstrating efficacy, but also safety. And then that first needs to be done in a number of animal studies before you can get to human trials. Now, when you talk about antibiotics that work against really dangerous pathogens, there is some opportunity for this process to be accelerated, especially if you're talking about maybe a new antibiotic that works against bacteria that have become pan-resistant, for which there are no proper treatments. And in those cases, there are now initiatives that are being taken and have been implemented by the FDA in the United States to try and accelerate the approval process. But at the end of the day, you still need to first go through the clinical trial process to demonstrate safety and efficacy in humans. And that is an expensive and time-consuming process. There are not that many companies chomping at the bit to invest in this type of development work for antibiotics because of the uncertainty associated with their being able to turn a profit or recoup their costs at the end if they bring a new antibiotic to market. So there's the framework is there for how to do it, but as long as the economics remain so dismal, it's hard to really predict how quickly a promising compound that comes out of my lab today would actually ever reach patients. So this is a big question mark, and I think governments around the world actually need to step up and think about how they can address this financial barrier to antibiotic development, because the science is there. I mean, we are doing good stuff, but certainly the best people to discover new antibiotics would be the people working at drug companies, the professional drug hunters. If they are properly incentivized to do so, they will be able to develop new antibiotics. I believe that. I think as an academic scientist, what we can do is sort of keep things going, train young scientists so that if and when companies are properly incentivized to work in this field, there'll be also qualified researchers to do those jobs. And of course, the other thing that academic science is very good at is understanding processes and mechanisms of disease and of resistance. So as I mentioned, it all actually does start with understanding how a bacteria becomes resistant. And if you understand that, then you can start imagining ways to overcome that resistance. So I think there's of course always going to be a role for academic researchers like myself. But I do worry about the lack of incentives for companies to work in this space. And I think until that's properly or effectively addressed, it's very difficult to imagine timelines for new antibiotics coming to the market. And certainly in the meantime, bacteria aren't waiting. They're continuing to evolve their resistance mechanisms to our existing class of antibiotics. So as you say, there are some reasons to feel rather dismal about the prospects here, unless we can sort of coordinate our efforts internationally and create some global incentives for companies to work in this space.
Do you have any funding issues or other problems to get new compounds or products validated?
Well, I mean, funding is difficult for everybody, ourselves included. And it's something of a rat race, if we're honest. I mean, everybody thinks their research is the most important thing. And I think a lot of people acknowledge that antibiotics research is important, but I'm competing with scientists who are, also interested in trying to develop effective medicines against cancers for which there are no good treatments, or for other chronic diseases. And because there are not big companies available to collaborate with on antibiotics, it's actually the case, I think, that it's rather difficult to find funding compared to other disease areas where there are huge profit motives. If you look at, for example, research in certain emerging cancer therapies, there's all kinds of funding available there. Fact is, though, if you don't have proper antibiotics backing up your healthcare system, a lot of innovative chemotherapies are very dangerous to use. A lot of people don't recognize that. But when you are treated with chemotherapy, oftentimes you're also treated with an antibiotic, sort of as a protective effect, because many chemotherapies can deplete one's immune system. So you need to also then take a prophylactic antibiotic. And without effective antibiotics, these, fancy and amazing new cancer medicines actually start to come with some increased risks. Same goes for a lot of other hospital procedures, by the way. So I think this is eventually a scenario that is going to start to bite us from multiple sides. And so definitely there should be more funding available for people doing the innovative discovery and development work. And to be honest, that's a relatively small amount of money compared to what it's going to take to really properly incentivize companies to want to do the later stage development work. So I would love to see more funding coming in for those of us that are doing the antibiotic discovery at the early stage. That's obviously essential. You can't develop an antibiotic candidate that hasn't yet been discovered. So yes, I would love to see more funding come in that direction. There are initiatives, but I think we need to do more. And I believe also if companies are properly incentivized to get back into this space, then it stands to reason that they would also then look to work together with research groups and other small companies interested in developing antibiotics. So there would be more resources from that side as well.
I think it's quite surprising. We're talking about a lot of money, perhaps billions of dollars. But on the other hand, if we're talking on a global scale, on a government level, it's not that much, especially when you consider the impact. It's such a pressing issue, and yet apparently it's still lacking urgency.
No, I think COVID really kind of shines a light on this too, right? Because we're spending trillions, globally on just measures to try and contain or repair the damage to the economy and the society that COVID's done. If we invest a few billion earlier on, you may be able to avoid that later on. So I think there might, my hope anyways, is that coming out of the COVID situation, people might also acknowledge, okay, let's appropriately invest and provide some incentives to develop new antibiotics now, rather than 10 or 20 years from now when the problem is really out of hand and the costs are astronomical.
Yeah, so this will maybe make us more willing to invest in prevention.
We do this for other things. I mean, the classic example would be fire extinguishers or fire trucks. We don't wait until a house is on fire before we go buy a fire extinguisher or build a fire truck. We have those things. We've invested in those things up front. We hope we never need to use them. But when they're necessary, we have them at our disposal, and you call the fire service, and in 2 minutes they're at your house to put your fire out before your house is burned to the ground, or the fire extinguisher can put the fire out in the kitchen before your whole house is on fire. Think of having a proper antibiotic arsenal in our hospitals like that to prevent future outbreaks of really difficult to treat antibiotics, being able to treat them when there's a small number of people who are infected rather than when they spread and get out of control.
Yes, so this will maybe make us more willing to invest in prevention. So in order to wrap up, what stage of development are you currently in?
So as an academic research group, we're very fundamental, very early stage. We would say discovery stage and maybe preclinical would be the two areas that we're able to get to. This is driven largely just by the costs that are involved in drug discovery and development. So the place we try to get to in our research is demonstrating efficacy in a relevant animal model. And if we can do that and convince ourselves and others that there's promise, then the idea or the hope is that outside parties would be then interested in licensing these discoveries. And then of course you get involved with the technology transfer offices at the universities, and they come into play too. So this, as I say, the discovery, the actually design and synthesis of the new antibiotics, and then we can do what are called in vitro tests in the lab. That's simply where you see now, do these new antibiotics kill drug-resistant bacteria in a Petri dish or in a 96 well plate? So we do that every day. There's people in the lab right now doing those types of experiments on new antibiotics. And if they work well in a 96 well plate, then we can escalate, as I say, up to an animal model of infection. And one of the benefits actually of working in an area like antibiotic research is the animal models are fairly predictive for how well you might expect a new antibiotic to work in higher animals or in humans. This is one of the things that actually makes antibiotic research, I won't say easier, but certainly the models are more predictive. And so we try as much as we can to validate our compounds up to the point where we see that they work well, for example, in a mouse model of infection.
Professor Nathaniel Martin from Leiden University. Thank you for taking the time to tell us more about your research and its importance in staying ahead of AMR. In our search to understand and overcome the resistance mechanism that bacteria develop, we could, like Professor Martin is doing, focus on the most important components on a molecular level and see if we can manipulate them or overcome them. This is a more analytical approach of the issue. Another possibility is to go even more fundamental by focusing on the mechanism that causes evolution in the first place. So instead of merely looking at, let's say, binding receptors and finding solutions to resensitize them, we focus on the evolutionary process of natural selection. My last guest is Professor Arjan de Visser. He's an evolutionary biologist at the Wageningen University. Welcome, Dr. de Visser. Could you please briefly introduce yourself?
My name is Arjan de Visser. I'm a biologist, an evolutionary biologist, and we work on models to try to predict evolutionary pathways. So, and for that, I'm not a modeler, so I work with people who are theoreticians that build the models, but we do the experiments. And the way we do this is we use bacteria and fungi in so-called laboratory evolution experiments to try to understand exactly what are the determinants of evolution. So what determines that bacteria evolve in a certain way?
You try to predict the evolutionary pathways of bacteria. Could you tell me why this is relevant for the AMR topic?
Well, this is relevant, we hope, because if we can predict how bacteria evolve resistance, we may be able to control this process better by designing therapies that then can counter, say, the evolution that we predict that will occur.
We just spoke with Dr. Martin about the several mechanisms bacteria develop to defend themselves against antibiotics. One of them is creating enzymes that will attack the medication. And you try to understand at a very fundamental level the genetic evolution of this enzyme. So actually, it's not the bacteria itself that mutates, but it's his weapon, the enzyme, the beta-lactamase.
Exactly. Yeah. So this is one approach we take. So there we focus on the evolution of the beta-lactamase and becoming more resistant to this new antibiotic.
Okay, so this is one of the two focus areas. But first, about the antibiotic you use in your experiment. Is that an antibiotic that is commonly used?
Yeah, so the beta-lactam that we use typically is cefotaxime. That's a so-called third-generation cephalosporin. So this is one member of this big group of beta-lactam antibiotics that started with penicillin in the 1940s, and now it's and then later synthetic versions of penicillin have been developed by chemists. And so this class of antibiotics, this beta-lactam group of antibiotics, is the most widely used group of antibiotics worldwide.
So beta-lactam is the antibiotic, and the beta-lactamase is the enzyme which is produced by the bacteria that kills the antibiotic. So could you tell me how you try to understand the determinants of the evolution and therefore try to predict its evolution path?
We do that in two different ways. So our focus is on the evolution of beta-lactam resistance. Beta-lactam is the largest used group of antibiotics worldwide. And we study the evolution of beta-lactam resistance in two different ways. The first way is a very simple and controlled way, where we only focus on one mechanism that leads to resistance to the beta-lactams that we use, where we only focus on the evolution of the enzyme involved in the breakdown of this antibiotic, called the beta-lactamase. And this we do by by introducing mutations in the gene for the enzyme in a PCR machine, so outside the bacterium.
And what is a PCR machine?
A PCR machine is a machine that copies DNA. And so we put the original gene, the DNA of the gene for the beta-lactamase in the machine. We make copies. The copies contain mutations. And so some of these mutations cause the enzyme to work better in breaking down the new antibiotic. So that is called in vitro evolution.
Okay.
And then we put the mutated versions of the gene back into the bacteria. We challenge them with the new antibiotic, and we select for resistance. And this way we can see the pathways where the enzyme is involved in resistance to the beta-lactam, and how many there are, and what the influence of environmental factors is on the choice of these different pathways. But we also have a broader approach where the enzyme is just one of the possible mechanisms of resistance to the beta-lactam antibiotics. And for that, we use a more conventional method by putting the gene for the enzyme on a plasmid in the strain of E. coli that we use, and we challenge both the plasmid and the chromosome with all the other possible mechanisms that may lead to resistance with the same antibiotic. And now we look at the whole broad range of pathways involving different mechanisms of resistance. And there we are interested in the same questions: how many different pathways there are, and what makes certain bacterial populations choose for one pathway over the other.
Let me try to summarize that. So the first angle of research is the most fundamental and solidly focuses on the pathways of the enzymes, which is the main mechanism bacteria use to evolve to resistance. Then we have the second research angle, which is broader. That research recognizes enzymes as being part of a broader biological mechanism, a part of the whole bacteria, which as a whole mutates as well. Then my next question is, what are the external factors that cause mutations of enzymes into certain directions?
Mutations occur just spontaneously when bacteria copy DNA. It's also true that in an environment where the antibiotic, the beta-lactam, is present, mutation rates may slightly go up because the antibiotic itself is somewhat mutagenic. But in terms of which of these mutations then survive and determine the route towards resistance that the population takes, we are particularly interested in the effects of population size, how many mutations are present in a population from which the population can choose. That's one very general factor we're interested in. And the other is how the different resistance mutations that the bacteria may generate affect the fitness of the bacteria themselves. So how does it affect their growth rate, if you want, because it's by natural selection that these mutations get selected, but they're not selected for the resistance effects directly. They're selected for their effects on the fitness, the survival and reproduction rate of the bacteria. That causes them to become dominant or not.
Do we already have enough understanding of the evolution process? I actually mean, can you already reproduce the same paths of mutations?
You mean that we see results in our experiments back in, say, clinical isolates? That is indeed what we see with the in vitro, with the simple version of our evolution experiments, the in vitro evolution of TEM1 beta-lactamase. So there we repeatedly find in our experiments an allele with 3 mutations that are exactly the same mutations as in TEM52, an isolate that is found in many clinical or a version that's found in many clinical isolates. So there apparently we are able to repeat natural evolution of this beta-lactamase. But in general, this is a long-term perspective to try to predict exactly what's going on in nature, if you want, in the clinic. So we are mainly focusing now on trying to find organizing principles that with some general applicability. For instance, the role of population size in the choice of different resistance mechanisms, having to do with what drives these different mechanisms. Are these high rate mutations or high benefit mutations for the bacterium? And so with that, if we find this for a certain antibiotic, this may apply to other mechanisms as well, so also to other antibiotics, we hope. So this may be relevant for understanding the natural evolution of resistance also towards these other antibiotics.
Will these research insights contribute in optimizing the use of existing drugs, or will it help in the design of new ones?
Well, that's a long haul. So one principle that people are investigating and that we could contribute to is this principle of collateral sensitivity. So this is what occurs when the bacterium becomes resistant to drug A and thereby becomes more sensitive to drug B. So if you know this is happening, we can, with drug A, we can, of course, try to predict an effective therapy with the combination of these two drugs, where you first apply A and then B to kill off the resistant bacteria to A.
So it's some kind of manipulation of the direction of its evolution path.
It's it's basically trying to understand how bacteria deal with these different resistance mechanisms that are available. So if resistance to one drug comes with higher sensitivity to another drug, we can of course exploit this. And with our evolution experiments and the protocols we develop, we have tools now to kind of analyze that at large scales. So we can, for instance, we use small droplets in which we put bacteria that are challenged with antibiotics. We use pipetting robots to transfer many populations of bacteria in parallel. And so these large-scale experiments are really very good at exploring a whole range of conditions and antibiotics for their effects that they may have also on the sensitivity to other drugs, so interactions between drugs.
Of what I understand, most of the experiments are conducted manually. Trying to truly understand the evolution path means that one is able to repeat the exact same path of mutations over and over again. If this is done manually, then it sounds like a very repetitive procedure. Isn't there a way to automate this process?
Yes, that's indeed a very a big challenge now. So we indeed want to get an overview of the mutational pathways towards resistance, and thereby we want to explore them as broadly as we can. So with very high replication of our experiments, and we use two different tools for that. So in one tool we have bacteria evolving in small droplets inside a machine. So these bacteria are in a small tube filled with oil. The bacteria are then in small water droplets in this oil. They're pumped back and forth, and they're detected by the fluorescent markers that they have, so we can see how many there are in each droplet. And they're challenged with antibiotics, and so we can repeat the growth process within these droplets by getting them out and putting small fractions back into new droplets again. And so this is one machine to look at the highly replicated evolution of bacteria challenged with antibiotics. And another tool we use in collaboration with the group in Cologne, at University of Cologne in Germany, is a pipetting robot where we evolve bacteria in so-called microtiter plates in small volume, sitting in small dents into a big plate, and they are transferred automatically by this robot. So this is another tool to study the highly replicated evolution towards antibiotic resistance.
How common is this research topic, focusing on the most fundamental aspect of a disease? Are there other research groups researching this as well, or is there any international collaboration on the topic?
The topic of the predictability of evolution is certainly one that is receiving growing attention. So we are part of a bigger consortium at the university that's coordinated at the University of Cologne in Germany, which is a mix of biologists, physicists, and medical people that have united to work on this problem of the predictability of evolution. And so we focus on the predictability of antibiotic resistance evolution, but others focus on the predictability of cancer evolution or the evolution of the immune system there. And so this is a mixture of experimentalists and theorists that then build models based on our experiments to try to predict the evolution of all kind of different unwanted processes like antibiotic resistance, cancer, but also the evolution of new pathogens such as influenza or currently COVID.
Thank you, Dr. de Visser. Thank you for your time. And I also want to thank my other guests, Dr. Sam Linsen of Squared End and Dr. Nathaniel Martin from Leiden University. More information on the speakers and their research can be found on our website, netherlandsinnovation.nl. Here you can also leave a comment. If you would like to reach us, please send an email to china@netherlandsinnovation.nl. With this episode, we finish the topic of AMR for now, and in our next episodes, we will focus on other Sino-Dutch technologies and innovations. Hope you'll be tuning in again, and thank you for listening.